And my research is mainly in the nanomedicine field, which is applying nanotechnology to address medical problems. So most recently, we have been working on is using magnetic nanoparticle for bioimaging, drug delivery, and drug screening.
And what we are doing in my lab, we are trying to find new drugs using innovative technologies, and we are screening very complex samples for the presence of possible drug leads that can be used in the prevention or treatment of aging-associated diseases.
The goal of our research is to identify the exact molecular mechanisms that take place with aging in the process of neurodegeneration. So in our research, we use the fruit fly Drosophila as a model organism to investigate the mechanisms of neurodegeneration. Flies serve as a great tool for our studies because they share many of the molecular and cellular pathways with higher organisms, including humans. And in addition to that, there is a great source of available genetic and molecular tools that allow us to perform experiments to look for neurodegeneration and different behavior changes or changes in lifespan or markers of inflammation that are typically seen in neurodegenerative diseases in humans as well.
I personally have been interested in natural products or compounds produced by plants, fungi, and bacteria present in the nature. And not many people actually realize how many drugs that we are currently using, even in the clinics, actually were first identified in nature. The problem that we usually have when we are working with natural products is unfortunately the fact that plants, fungi, or bacteria produce a lot of different molecules. And it is always very difficult to tell the difference between what is pharmacologically or biologically active from something that is not going to be active. And here is where our technology comes. It actually allows us to speed up the process of identification, to tell the difference between the molecules that will be useful as a potential drug and those that will not work.
So it's mainly the design part is our bait, is the receptor on the cell we're using. So we're only going to be like a smart bait, only going to fish out the one bind to our target, our interest.
What we would like to investigate is how the elevated levels of antimicrobial peptides, specifically in the brain, lead to neurodegeneration. And the funding from the Merrymac-McKinley Foundation will allow us to perform some preliminary experiments in a way to address this question.
And this Merrymac-McKinley support will really help us to generate some preliminary results. So this McKinley support is really just focusing on us to say, let's generate the first set of preliminary results, get the paper published, so we can convince people saying that this technique is really working, will have a lot of potential, so lead to the big awards from mainly NIH.
This Merrymac-McKinley Foundation funding will allow us to generate data that will show the broader applicability of our technique and its usefulness, and it will also allow us to validate the entire process. So Alabama Life Research Institute is interested in one of the main topics is also rural health, and it has been shown that a lot of diseases, including neurodegenerative diseases, are actually more prevalent in rural areas. That's actually, for example, true to Parkinson's disease. And if you look at the map of the entire United States, the southern states actually tend to have the higher prevalence of neurodegenerative diseases. So our research that focuses on identification of possible new drug leads falls well within the broad scope of interest of Alabama Life Research Institute.